Chapter 14, IgE-Mediated Immunity and Allergy. Different effector mechanisms cause 4 distinctive types of hypersensitivity reaction. This figure explains the 4 major types of hypersensitivity reactions, each involving different immune mechanisms. Type I hypersensitivity begins when an antigen binds to immunoglobulin E that is attached to receptors on the surface of mast cells. This binding triggers the release of inflammatory mediators from the mast cells, leading to allergic reactions such as hives, asthma, or anaphylaxis. Type 2 hypersensitivity occurs when immunoglobulin G or immunoglobulin M antibodies bind to antigens that are present on the surface of host cells. This can happen, for example, when penicillin modifies host proteins and creates new epitopes. The binding of antibodies triggers complement activation and the recruitment of macrophages, leading to the destruction of the targeted cells. Type 3 hypersensitivity involves the formation of immune complexes between antibodies and soluble antigens. These complexes can deposit in tissues such as blood capillaries, where they activate complement and attract neutrophils. The resulting inflammation and tissue damage can cause diseases such as serum sickness and vasculitis. Type 4 hypersensitivity is a delayed type reaction that is mediated by T cells rather than antibodies. For example, in response to metals like nickel introduced through body piercings, antigen-presenting cells migrate to the lymph nodes and activate T cells. These activated effector CD4 T cells return to the site of exposure days later and cause tissue damage through macrophage activation and inflammatory cytokine production. This figure shows that although all hypersensitivity reactions involve inappropriate immune responses, the specific immune mechanisms differ significantly between the 4 types. IgE-mediated immune responses defend the body against multicellular parasites. This figure describes the major components of a typical adaptive immune response against helminth worms, which are multicellular parasitic organisms. The T cells involved in this response are CD4 T helper type 2 cells. These cells play a central role in coordinating the immune attack against large parasites that cannot be easily destroyed by phagocytosis. The cytokines that mediate the response include interleukin-3, interleukin-4, interleukin-5, interleukin-9, interleukin-10, and interleukin-13. These cytokines help stimulate the activation and recruitment of various immune cells that are effective against helminth infections. The antibodies produced during the response include immunoglobulin E, immunoglobulin G1, and immunoglobulin G4. Immunoglobulin E, in particular, is crucial for arming mast cells, basophils, and eosinophils to attack the parasites. The effector cells involved in the response include expanded populations of eosinophils, basophils, and mast cells. These cells release toxic mediators that can damage and expel helminth worms from the body. This figure highlights how a coordinated effort involving T cells, cytokines, antibodies, and effector cells is required to effectively combat large multicellular parasites. This figure shows a world map highlighting the regions where parasitic diseases are most prevalent. The areas shaded in red represent countries and regions where infections caused by parasitic organisms, including helminths and protozoa, occur frequently and pose major public health challenges. Parasitic diseases are especially common in sub-Saharan Africa, where multiple parasitic infections, including malaria, schistosomiasis, and various intestinal helminth infections, are endemic. Significant portions of South America and Central America are also heavily affected by parasitic diseases, particularly in tropical and subtropical climates where sanitation infrastructure may be limited. In South Asia, countries such as India and Bangladesh show high prevalence of parasitic infections, as do parts of Southeast Asia and the Western Pacific Islands. Scattered regions in the Middle East and North Africa are also shown as areas with significant parasitic disease burden, as are parts of Central Asia. In contrast, most of North America, Europe, and Australia are shown with a lower prevalence of parasitic infections, reflecting differences in public health systems, sanitation, and access to healthcare resources. This map emphasizes that parasitic diseases remain a major cause of illness and death in many parts of the world, particularly in regions where access to clean water, sanitation, and medical care is limited. IgE antibodies emerge at early and late times in the primary immune response. This figure shows the organization of immunoglobulin heavy chain June segments and explains how immunoglobulin E antibodies emerge during the course of a primary immune response. The diagram represents the constant region June segments that encode the different immunoglobulin isotypes. At the beginning of the locus are the genes encoding the heavy chain constant regions for immunoglobulin M and immunoglobulin D. Following these are two duplicated sets of gene segments, known as duplication unit 1 and duplication unit 2. Each duplication unit contains genes encoding constant regions for immunoglobulin G subclasses, immunoglobulin A subclasses, and immunoglobulin E. Within duplication unit 1, the June segments include immunoglobulin G3, immunoglobulin G1, an inactive immunoglobulin E gene, and immunoglobulin A1. Duplication unit 2 contains an inactive immunoglobulin G Jones segment, immunoglobulin G2, immunoglobulin G4, immunoglobulin E, and immunoglobulin A2. During a primary immune response, immunoglobulin E antibodies can be produced early if class switching occurs to the inactive immunoglobulin E Jones within duplication unit 1. Alternatively, immunoglobulin E antibodies can be produced later if class switching proceeds to the active immunoglobulin E Jones found in duplication unit 2. This arrangement explains why immunoglobulin E can appear both early and late during an immune response, depending on which Jones segment is targeted during class switching. This figure presents data comparing the mean number of somatic mutations in the heavy chain variable regions of different antibody isotypes. Somatic mutation is a process that introduces small genetic changes into the variable regions of immunoglobulin genes. These mutations contribute to the process of affinity maturation, allowing the immune system to produce antibodies with increased binding strength over time. The table shows that immunoglobulin E antibodies have the fewest somatic mutations, with an average of 12 mutations in the heavy chain variable region. This suggests that immunoglobulin E antibodies often arise earlier in the immune response, before extensive rounds of mutation and selection occur. In contrast, immunoglobulin G subclasses show progressively higher numbers of somatic mutations. Immunoglobulin G3 antibodies have an average of 18 mutations. Immunoglobulin G1 antibodies average 21 mutations. Immunoglobulin G2 antibodies average 22 mutations. And immunoglobulin G4 antibodies have the highest mean with 27 mutations. This pattern supports the idea that immunoglobulin E antibodies can be produced either early or late in an immune response. Early immunoglobulin E responses likely have fewer somatic mutations, whereas immunoglobulin G antibodies, especially immunoglobulin G4, are produced later after prolonged affinity maturation. This figure emphasizes how the number of somatic mutations correlates with the timing and quality of antibody responses during an immune challenge. This figure illustrates how immune complexes containing different antibody isotypes regulate macrophage activation during an immune response. The diagram shows an immune complex that includes immunoglobulin G4 bound to an inhibitory receptor called Fc-gamma receptor IIB and immunoglobulin G1 bound to an activating receptor called Fc-gamma receptor IIA on the surface of a macrophage. The binding of immunoglobulin G1 to Fc-gamma receptor IIA sends an activating signal into the macrophage, promoting inflammatory responses. However, when immunoglobulin G4 binds to Fc-gamma receptor IIB, it sends a strong inhibitory signal that counteracts the activating signal. In this context, the inhibitory signals from Fc-gamma receptor IIB are stronger than the activating signals from Fc-gamma receptor IIA. As a result, the macrophage does not become fully activated and does not secrete inflammatory cytokines. This mechanism allows immune complexes containing immunoglobulin G4 to suppress inflammation by preventing macrophage activation, contributing to immune regulation and limiting tissue damage during chronic or excessive immune responses. Allergy is prevalent in countries where parasite infections have been eliminated. This figure explains the Hygiene hypothesis and its connection to the rise of allergic diseases in countries where parasite infections have been largely eliminated. According to the Hygiene hypothesis, excessive hygiene practices during childhood reduce exposure to both commensal and pathogenic microorganisms, as well as to other humans and animals. This limited exposure prevents the developing immune system from experiencing natural infections, confronting pathogens, and successfully terminating infections. Vaccination, while crucial in preventing serious diseases, also reduces the immune system's opportunity to practice fighting infections independently, decreasing its experience in recognizing and eliminating real threats. An over-reliance on antibiotics further limits the immune system's engagement with pathogens, weakening its ability to distinguish between self and foreign invaders. Antibiotic use can also promote the emergence of resistant bacteria, which complicates immune responses and further perturbs normal immune function. As a result, the immune system during childhood becomes poorly educated, under-exercised, and inexperienced in combating real infections. Consequently, the immune system may turn its activity toward fighting non-existent threats, leading to the development of debilitating allergic diseases. IgE has distinctive properties that contrast with those of IgE. This figure shows the distinctive structure of the immunoglobulin E antibody compared to other antibody classes. Immunoglobulin E is uniquely designed for high-affinity binding to receptors on immune cells. The immunoglobulin E molecule is composed of 2 heavy chains and 2 light chains. Each heavy chain includes 4 constant regions, labeled constant epsilon-1, constant epsilon-2, constant epsilon-3, and constant epsilon-4. These constant regions give immunoglobulin E its extended and rigid structure. The immunoglobulin E antibody binds to the high-affinity receptor for immunoglobulin E, which is found on mast cells and basophils. This receptor, known as the high-affinity immunoglobulin E receptor, is made up of 1 alpha chain, 1 beta chain, and 2 gamma chains, which transmit activation signals inside the cell through immunoreceptor tyrosine-based activation motifs. The structure of immunoglobulin E and its specialized receptor enables a rapid and powerful immune response upon antigen exposure, a feature that distinguishes it from immunoglobulin G and other antibody classes. IgE and FcεRI supply each mast cell with a diversity of antigen-specific receptors. This figure illustrates how B cells and mast cells interact with a diversity of antigens through specific receptors. At the top, 2 panels show B cells. Each B cell expresses one type of receptor specific for a particular antigen. One B cell displays receptors that recognize herringworm antigens, while another B cell displays receptors that recognize pollen antigens. Each B cell is committed to a single specificity. At the bottom, the mast cells are shown. Unlike B cells, mast cells can be loaded with immunoglobulin E antibodies of many different specificities. These immunoglobulin E molecules are bound to high-affinity receptors for immunoglobulin E on the mast cell surface. As a result, a single mast cell can simultaneously display receptors specific for herringworm, blood fluke, hookworm, and roundworm. Another mast cell may carry antibodies against pollen, cat dander, peanut allergens, and bee venom. This diversity equips mast cells to respond rapidly and broadly to a wide range of environmental threats. This figure illustrates how mast cells are armed with a wide diversity of antigen-specific receptors through the binding of immunoglobulin E to high-affinity immunoglobulin E receptors. Each immunoglobulin E molecule recognizes a different antigen. These immunoglobulin E molecules are bound to the high-affinity receptor for immunoglobulin E, which is composed of alpha, beta, and 2 gamma chains. When an antigen binds to 2 adjacent immunoglobulin E molecules on the mast cell surface, it triggers cross-linking of the receptors. This cross-linking initiates a signal inside the mast cell, leading to its activation. Activated mast cells then release the contents of their granules, such as histamine and other inflammatory mediators, into the surrounding tissue. This release promotes a rapid immune response against parasites or allergens. FcεRII is a low-affinity receptor for IgE regions that regulates the production of IgE by B cells. This figure compares 2 different receptors for immunoglobulin E on the left. The high-affinity receptor for immunoglobulin E is shown. This receptor consists of alpha, beta, and 2 gamma chains and binds immunoglobulin tightly to the surface of cells such as mast cells and basophils. On the right, the low-affinity receptor for immunoglobulin E is shown. This receptor, called the low-affinity receptor for the fragment crystallizable region 2, binds immunoglobulin E less tightly and is expressed mainly on B cells. The low-affinity receptor for the fragment crystallizable region 2 plays a regulatory role by helping to control the production of immunoglobulin E by B cells. When immunoglobulin E binds to this receptor, it can provide feedback signals that limit further immunoglobulin E production. This mechanism helps prevent excessive allergic responses by regulating the amount of immunoglobulin E produced in the body. This figure shows how the low-affinity receptor for the fragment crystallizable region 2, or FcεRI receptor 2, regulates the production of immunoglobulin E by B cells. The receptor cross-links the B cell receptor and the B cell co-receptor. This interaction triggers signals that lead to the activation and differentiation of the B cell. As a result, the B cell transforms into a plasma cell that specifically secretes immunoglobulin E. This mechanism helps ensure that immunoglobulin E production is tightly regulated during the immune response, especially during allergic reactions or parasitic infections. Treatment of allergic disease with an IgE-specific monoclonal antibody. This figure shows 2 different effects of monoclonal antibodies that target immunoglobulin E. On the left, therapeutic anti-immunoglobulin E antibodies prevent mast cells from acquiring immunoglobulin E on their surface. Without surface immunoglobulin E, mast cells cannot be activated through the high-affinity receptor, and surface expression of the receptor is reduced. On the right, experimental anti-immunoglobulin E antibodies bind to immunoglobulin E already attached to the high-affinity receptor. This cross-linking activates mast cells, leading to their deregulation and triggering an inflammatory response. This highlights the importance of carefully designing anti-immunoglobulin E therapies to avoid unintended activation of mast cells. Mast cells defend and maintain the tissues in which they reside. This image shows a mast cell highlighted in red among surrounding tissue cells stained blue. Mast cells are long-lived, tissue-resident cells that play a key role in maintaining tissue health and defending against pathogens. They are loaded with granules that contain powerful mediators, which they release in response to injury or infection. By sensing their environment and responding rapidly, mast cells help protect and repair the tissues in which they reside. Mast cells release a variety of products to defend tissues. Enzymes such as tryptase and chymase remodel the connective tissue matrix. Toxic mediators like histamine and heparin poison parasites increase vascular permeability and cause smooth muscle contraction. Cytokines, including tumor necrosis factor alpha, promote inflammation, while interleukin-4 and interleukin-13 stimulate the T helper 2 cell response. Chemokines such as CCL3 attract monocytes, macrophages, and neutrophils. Lipid mediators like leukotrienes and platelet-activating factor cause smooth muscle contraction, increase vascular permeability, and recruit additional immune cells. These coordinated responses protect and maintain tissue integrity during infection and injury. Tissue mast cells orchestrate IgE-mediated reactions through the release of inflammatory mediators. Tissue mast cells orchestrate immunoglobulin E-mediated reactions by releasing inflammatory mediators, such as histamine. Histamine, derived from the decarboxylation of histidine, plays a central role in promoting vascular permeability, smooth muscle contraction, and recruitment of immune cells to sites of infection or injury. These effects help amplify local inflammation and contribute to allergic responses. Tissue mast cells release lipid mediators derived from arachidonic acid through the cyclooxygenase and 5-lipoxygenase pathways. Cyclooxygenase produces prostaglandin D, while 5-lipoxygenase produces leukotriene C, both of which contribute to inflammation by enhancing vascular permeability, smooth muscle contraction, and leukocyte recruitment. Aspirin inhibits prostaglandin synthase by acetylating the enzyme, thereby blocking prostaglandin production and reducing inflammation. Eosinophils are specialized granulocytes that release toxic mediators in IgE-mediated responses. Eosinophils are specialized granulocytes that participate in IgE-mediated immune responses by releasing toxic granule proteins. These cells are characterized by their bilobe nuclei and prominent cytoplasmic granules, which contain enzymes and proteins capable of damaging pathogens and contributing to tissue inflammation during allergic reactions. Eosinophils contribute to immune defense through the release of a variety of products. Their enzymes, including eosinophil peroxidase and eosinophil collagenase, help poison parasites and remodel connective tissue. Toxic proteins such as major basic protein, eosinophil cationic protein, and eosinophil-derived neurotoxin target parasites and mammalian cells, trigger histamine release, and degrade RNA. Cytokines like interleukin-3, interleukin-5, and granulocyte macrophage colony-stimulating factor promote eosinophil production and activation. Chemokines like CXCL8 attract leukocytes, while lipid mediators, including leukotrienes and platelet-activating factor, enhance vascular permeability, smooth muscle contraction, mucus secretion, and immune cell recruitment. Basophils are rare granulocytes that initiate tissue responses and the production of IgE. This image shows a basophil among surrounding red blood cells. Basophils are rare granulocytes that play a crucial role in initiating T helper 2 or Th2 responses and promoting the production of immunoglobulin E or IgE. Their granules, stained dark purple, contain mediators that help drive allergic and antiparasitic immune responses. Allergens are protein antigens, some of which resemble parasite antigens. This chart explains features of inhaled allergens that promote priming of T helper 2 cells that drive the immunoglobulin E response. Allergens are proteins because only proteins can induce T cell responses. Many allergens act as proteases. A low dose favors activation of interleukin-4 producing CD4 T cells. Allergens with low molecular mass easily diffuse out of particles into mucus. High solubility allows allergens to be readily eluted from particles. High stability enables allergens to survive in dry particles. Finally, allergens that contain peptides binding to host major histocompatibility complex class II molecules are necessary for T cell priming. This diagram illustrates the process of allergic sensitization. During the first exposure to pollen, allergens are inhaled into the airway. Antigen-presenting cells extract and process the antigen. These cells then activate antigen-specific T cells. Activated T helper 2 cells stimulate B cells to produce immunoglobulin E, which binds to Fc receptors on mast cells, priming them for future allergic reactions. Certain helminth antigens closely resemble allergens that trigger immune responses in humans. The herring worm produces cystidine, cytochrome C oxidase subunit 3, and troponin C, which are similar to allergens for minor cat dander, Bermuda grass pollen, and German cockroaches. Blood fluke antigens, such as cathepsin B like cysteine protease, share similarity with the papain allergen. Roundworm produces glutathione S-transferase, related to dust mite allergens. Malayan filaria expresses tropomyosin, which matches the same allergen found in environmental exposures. Predisposition to allergic disease is influenced by genetic and environmental factors. Genetic polymorphisms influence susceptibility to allergic diseases by affecting immune system regulation. Structural variants in MHC class II genes enhance the presentation of allergen-derived peptides. Non-coding variants in the T cell receptor alpha locus increase T cell recognition of allergens. Promoter and structural variants in the TIM-Jun family regulate the balance between Th1 and Th2 responses. Variants in the IL-4 and IL-4R genes alter the expression and signaling pathways of IL-4, a key cytokine in allergic responses. Changes in the high-affinity IgE receptor beta chain impact IgE-mediated activation. Variants in ALOX5 influence leukotriene production, affecting inflammation. Mutations in the beta-2 adrenergic receptor increase bronchial hyperreactivity, while changes in ADAM33 contribute to variation in airway remodeling. In allergic responses, antigen-presenting cells capture allergens and present them via MHC class II molecules to naive T cells. Genetic variations in MHC class II can enhance the presentation of allergen peptides. The activated T cells differentiate into Th2 cells, which secrete cytokines that stimulate B cells. Upon activation, B cells undergo class switching to produce IgE antibodies. The produced IgE binds to FcεRI receptors on mast cells and basophils. Genetic polymorphisms in receptors, cytokines, and associated pathways modulate these interactions, influencing the severity and progression of allergic diseases. IgE-mediated allergic reactions consist of an immediate response followed by a late-phase response. During an allergic response, the immediate phase occurs within minutes of allergen exposure and is characterized by redness, swelling, and itching due to the rapid release of histamine and other performed mediators from mast cells. Several hours later, a late-phase response develops, marked by sustained inflammation and tissue swelling, as newly recruited inflammatory cells, such as eosinophils and basophils, release additional mediators that prolong and intensify the allergic reaction. Upon antigen challenge, there is a rapid and sharp decline in PEFR within the first 30 minutes, indicating bronchoconstriction typical of the immediate phase reaction. This immediate drop is followed by a recovery back to baseline levels after about an hour. However, after several hours, a second, more prolonged decrease in PEFR occurs, marking the late phase reaction. This late phase bronchoconstriction can last for several hours, demonstrating the sustained inflammatory response characteristic of IgE-mediated allergies. The effects of IgE-mediated allergic reactions vary with the site of mast cell activation. In the gastrointestinal tract, mast cell activation leads to increased fluid secretion and peristalsis, causing diarrhea and or vomiting to expel gastrointestinal contents. In the airways, activation results in decreased airway diameter and increased mucus secretion, leading to coughing, sneezing, and phlegm expulsion. In the blood vessels, mast cell activation causes increased blood flow and vascular permeability, promoting edema, inflammation, and enhanced flow of antigens to lymph nodes. Systemic anaphylaxis is caused by allergens in the blood. When an antigen enters the bloodstream, it activates IgE-coated mast cells throughout the body. Upon degranulation, these mast cells release inflammatory mediators that affect multiple systems. In the heart and vascular system, there is increased capillary permeability and fluid leakage into tissues, leading to swelling, loss of blood pressure, reduced oxygen delivery, irregular heartbeat, anaphylactic shock, and potential loss of consciousness. In the respiratory tract, smooth muscle contraction and airway constriction cause difficulty swallowing, breathing, and wheezing. In the gastrointestinal tract, smooth muscle contraction leads to stomach cramps, vomiting, increased fluid outflow, and diarrhea. Following antigen exposure time marked by arrows, there is an immediate sharp decrease in mean arterial pressure red line and a rapid spike in plasma epinephrine levels blue line. The body's natural release of epinephrine temporarily restores blood pressure toward normal. However, after this brief recovery, both blood pressure and epinephrine levels decline again, requiring clinical intervention if untreated. This graph highlights the urgency of recognizing and treating anaphylaxis quickly, often with epinephrine administration. Penicillin first binds to bacterial transpeptidase enzymes, inactivating them by forming a covalent bond through its beta-lactam ring. However, penicillin can also react with proteins on human erythrocytes using the same reactive beta-lactam bond. This chemical modification creates new, foreign epitopes on the surface of erythrocytes. The immune system then recognizes these altered proteins as antigens, potentially initiating an allergic response, including severe reactions such as systemic anaphylaxis. Complement-coated, penicillin-modified erythrocytes are first recognized and engulfed by macrophages. These macrophages then process and present peptides from the penicillin protein conjugate on MHC class II molecules, activating CD4 T cells to differentiate into Th2 cells. The activated Th2 cells, in turn, stimulate B cells that recognize the same antigen. These B cells differentiate into plasma cells that secrete penicillin-specific IgE antibodies. The IgE binds to FcεRI receptors on mast cells, sensitizing them. When penicillin-modified erythrocytes encounter these armed mast cells again, it triggers degranulation, leading to anaphylaxis. Rhinitis and asthma are caused by inhaled allergens. When an inhaled allergen enters the nasal mucosa, it activates local mucosal mast cells, This activation leads to increased blood vessel permeability and stimulation of the nasal epithelial cells. As a result, inflammatory cells, particularly eosinophils, are recruited from the bloodstream. These eosinophils migrate into the nasal passages, where they contribute to tissue edema and mucus production, ultimately causing symptoms of allergic rhinitis or asthma. Upon exposure to an inhaled allergen, mucosal mast cells in the airway rapidly capture the antigen. In the acute phase, inflammatory mediators are released, leading to contraction of the airway smooth muscle, increased mucus secretion by the airway epithelium, and heightened blood vessel permeability. These changes contribute to immediate symptoms such as airway constriction and mucus accumulation. In the chronic phase, continued inflammation is driven by the release of cytokines and eosinophil granule proteins. This ongoing response sustains tissue damage, airway remodeling, and persistent symptoms characteristic of chronic asthma. This figure shows histological changes associated with chronic asthma. In the left panel, staining highlights the thickened airway wall, including hypertrophy of smooth muscle and extensive collagen deposition, which leads to narrowing of the airway lumen. In the right panel, a dense infiltration of inflammatory cells, including eosinophils, is visible in the airway wall, contributing to persistent inflammation, tissue damage, and airway remodeling over time. Urticaria, angioedema, and eczema are allergic reactions in the skin. When a low dose of antigen enters the subcutaneous tissue, it activates mast cells in the skin. Upon activation, mast cells release mediators that increase vascular permeability, allowing cells, fluid, and proteins to move from the bloodstream into surrounding tissues. This leads to localized swelling known as wheal and flare reactions. Clinically, this process underlies conditions such as urticaria, angioedema, and eczema, all of which are characterized by redness, swelling, and itching in affected skin areas. Food allergies cause systemic effects as well as gut reactions. In food allergies, ingestion of an antigen activates mucosal mast cells in the gut. Activated mast cells release histamine, which acts on the intestinal epithelium, blood vessels, and smooth muscle. As a result, antigens diffuse into the bloodstream, causing systemic effects like urticaria. Meanwhile, smooth muscle contraction leads to vomiting and diarrhea. An increased vascular permeability causes fluid outflow into the intestinal lumen, contributing to additional gastrointestinal symptoms. Allergic reactions are prevented and treated by three complementary approaches. This figure compares immune system strength and disease outcomes between parasite-infested and parasite-free populations. In parasite-infested populations, a balance exists between tolerance, resistance, and immunopathology. Individuals with moderate immune strength resist parasites effectively, while those with weaker or stronger immune responses experience superinfection or immune-related tissue damage, respectively. In contrast, in parasite-free populations, strong immune responses shift toward allergic diseases rather than parasite control, increasing the prevalence of allergy in the population.